So remember back to the beginning of the last section on nonspecific resistance. There were 3 lines of defense against pathogens. The first line of defense consisted of these external barriers like the skin and mucous membranes. And the second line of defense was the And the second line of defense consisted of several non-specific defenses that we covered in our last section. Now we move on to specific immunity. In specific immunity, we deal with the immune system. It's composed of a large population of widely distributed cells that recognize foreign substances and act to neutralize and destroy them. There are 2 characteristics that distinguish immunity, specific immunity, from non-specific defenses or resistance. First of all, there's specificity. Immunity is directed against a particular pathogen. And then there's memory, in which when the immune system is re-exposed to that particular pathogen, the body reacts so quickly that there's no noticeable illness. There are 2 types of immunity. There's cellular-mediated immunity and humoral immunity. In cellular-mediated immunity, we see that the T cells are predominantly active. These T lymphocytes directly attack and destroy foreign cells or diseased host cells. This means the ridding of the body of pathogens that reside inside the cells. When we have pathogens that are located inside the cells, they're inaccessible to antibodies, so the response cannot be mediated by antibodies. This is one of the big problems with HIV. HIV enters the body and then it floats around in the blood for a short while, but then it hides out inside cells. Now, in its case, it becomes unrecognizable even when it's inside the cell, so our immune system really has a tough time attacking it. Humoral immunity is antibody-mediated immunity. It's mediated predominantly by the B cells. And antibodies, in this case, antibodies attack the enemy as it's floating around in the blood. They don't directly destroy the pathogen, but they label them for attack. The key here is the humoral response can only work against the extracellular stage of infectious microorganisms. The B cells can't help out once the pathogen enters a cell. So either type of immunity can occur naturally, or for treatment and prevention purposes, it can be induced artificially. So we can therefore recognize 4 classes of immunity under this scheme. We have natural active immunity, which is where we produce our own antibodies or T cells as a result of infection or natural exposure to the antigen. Then we can have artificial active immunity. This is a vaccine. We produce our own antibodies or T cells as a result of a vaccination against a disease. The vaccine consists of dead or attenuated, which are weakened pathogens. They stimulate an immune response without causing the disease. We'll have periodic booster shots to stimulate the immune memory to maintain a high level of protection. Then we have natural passive immunity. This is where we have temporary immunity that results from antibodies that are produced by another person. This happens really only when a fetus acquires antibodies from the mother through placenta and drinking of breast milk. This natural passive immunity is why it's so important for mothers to breastfeed, to pass on immunity that her cells have experienced in life to her fetus. In artificial passive immunity, we can see temporary immunity that results from injection of an immune serum, antibodies, that are made by either another person or animal. We see these in the treatment of snake bites, botulism, rabies, tetanus, and other diseases. We have an immune serum that goes in and, for example, picks up all the toxins of the snake bite and labels them for disposal by the body. Only the 2 forms of active immunity involve immune memory. and thus provide future protection. Both passive immunities don't have this effect. So pause here a moment and spend just 1 minute jotting down everything that you can remember that we've covered so far. Specifically, paying attention to the differences between cellular and humoral immunity, and then distinguish between passive and active. Just a quick exercise in recall. And now we'll move on to look at antigens. So an antigen is any molecule at all that triggers an immune response. They generally have large molecular weights. That's 10,000 atomic mass units in general. They're complex molecules with structures that are unique to the individual. They're made of proteins, polysaccharides, glycoproteins, glycolipids, and the body can distinguish self-antigen molecules from foreign antigen molecules, much like we observed in blood typing earlier in the semester. So antigen molecules are obviously quite large, and they only have certain regions that are going to be recognized by antibodies. This is the epitope region. It's the specific region of an antigen molecule that stimulates the immune response. Now, haptens are antigen molecules that are way too small to be antigenic themselves. Thus, they have to combine with a host's macromolecule in order to be recognized. Haptens are in things like cosmetics, detergents, industrial chemicals. They're in poison ivy and animal dander. Penicillin also has haptens. The haptens in penicillin bind to the host proteins, and then the host's immune system picks them up as an invader. Now let's look at the various lymphocytes. These are the major cells of the immune system. We've got lymphocytes, macrophages, and dendritic cells. They're especially concentrated, as we've seen, in specific places such as the lymphatic organs, the skin, and the mucous membranes. There are 3 categories of the lymphocytes. They're natural killer cells that we've seen already in immune surveillance. And now we'll get into more detail about T lymphocytes and B lymphocytes. As you'll recall, the T lymphocytes were involved in the cellular response, and the B lymphocytes are involved in the humoral response. So let's look at the life cycle of T cells to begin with. It involves 3 stages and 3 anatomical stations. They begin by being born in the red bone marrow. As you'll recall, they're descendants of PPSCs, or pluripotent stem cells, and they were released into the blood as still undifferentiated stem cells, and then they colonize the thymus. In the thymus, they're essentially in school or training. This is the site that they mature. The reticular epithelial cells secrete thymic hormones that stimulate the maturing T cells to develop surface antigen receptors. Those T cells are now immunocompetent. That is, they're capable of recognizing antigens presented to them by a school of cells called the Antigen-Presenting Cells. Now, in order to graduate from school, the T cells need to be tested. We need to be sure that they're able to recognize antigens, but not react to self-antigens. So the reticular epithelial cells will test the T cells by presenting self-antigens to them. They can fail the test in 2 ways. First of all, they may not recognize the reticular epithelial cells and their antigens. This means that they would be incapable of recognizing a foreign attack because they don't recognize antigens, period. Second, they could fail by reacting to a self-antigen. That means the T cells would always attack our own tissues. So this test is a process of negative selection. The T cells that fail the test need to be eliminated. They'll be eliminated by 2 processes. The self-reactive T cells could just die, and then macrophages would phagocytose them. This is called clonal deletion. Or they could go through the process of anergy. This is where self-reactive T cells remain alive, but they are not responsive. Only about 2% of immunocompetent T cells will be permitted to graduate. That is, only 2% will pass the test. If they do pass the test, they're allowed to move into the medulla. Here they multiply to form clones of identical T cells that are programmed to respond to specific antigens. At this point, they're a naive lymphocyte pool. They've graduated, but they haven't had any experience yet. They haven't encountered the foreign antigen that they're programmed to respond to. At this point, they'll be deployed. They'll leave the thymus and colonize lymphatic tissues and organs and await their first experience. B lymphocytes mature in a fairly similar way. However, as you know, they remain in the bone marrow. They develop into mature B cells there. They also go through a process of cell selection. B cells that react with self-antigens either undergo anergy or clonal deletion in a very similar way as T cell selection. Self-tolerant B cells will synthesize antigen surface receptors. They divide rapidly and they produce more and more immunocompetent B cell clones. They leave the bone marrow and colonize the same lymphatic tissues and organs as the T cells. So the B cells will be ready to attack anything that's floating around in the humor, in the blood. And the T cells will be ready to attack any cells that contain enemies. So stop here for a moment and diagram the process of T cell maturation. How they originate in the red bone marrow, then they're in the thymus, beginning in the cortex, where they're in school, testing happen, negative selection, positive selection, and then deployment to the lymphatic tissues. And now let's move on to look at antigen-presenting cells, the last piece of the puzzle that we're going to need to understand both cellular and humoral immunity. Antigen-presenting cells do exactly what they sound like. They present antigens. Because T cells can't recognize the antigens that they need to attack all on their own, they require the help of these antigen-presenting cells. Dendritic cells, macrophages, reticular cells, and B cells all act as antigen-presenting cells. The antigen-presenting cell function hinges on a family of proteins called major histocompatibility complex proteins, or MHC proteins. These MHC proteins act as cell ID tags, and they label every single cell in your body as belonging to you. The structure of these proteins is unique to every individual except for identical twins. So when an antigen-presenting cell encounters an antigen, as you can see here on the right, it internalizes it by endocytosis, digests it into molecular fragments, recognizes the epitopes, and then expresses them on the cell surface. Basically, it says to the T cells, hey, I've found some invaders, and this is what they look like. So then the wandering T cells will inspect all these antigen-presenting cells for antigens that they might recognize as foreign. If the antigen-presenting cell only displays self-antigens, well, then the T cell disregards it. But if it displays non-self-antigens, the T cell will initiate an immune attack. Thus, the antigen-presenting cell role is to alert the immune system of the presence of a foreign antigen. Because there are so many antigen-presenting cells, they're able to quickly mobilize immune cells against the antigen. With so many different cell types involved in immunity, they're going to require chemical messengers to coordinate their activities. This is where the leukotriene chemicals come in. Leukotrienes are signals that are released by leukocytes so that they can let each other know what's going on. So now that we've had a brief introduction to the general aspects of specific immunity, it's time to step into the cellular immunity.